These are things already we discussed about in yesterday class. Now we will discuss about today what is the beta oxidation. What is the oxidations are there, hydrations are there. Now we will discuss about to the only derived lipids. In the derived lipids, fatty acids, all are things in the yesterday class we will discuss, completely discuss about the yesterday class. Now, beta oxidation or fatty acid. Beta oxidations are fatty acid. Definition of beta oxidation may be defined as the oxidation of fatty acid, may be oxidation of the fatty acids on the beta carbon atom. What is that one? Beta carbon atom. This result in the sequential removal of this. You can observe this removal of the sequence and removal of this was removal of two carbon fragments. Two carbon fragments are there. In this one, two fragments is the acyl coenzyme A is present. Acyl coenzyme alpha beta oxidation and acyl coenzyme also to present in this process. Here, main is the beta oxidation in the where the carbon atom is there. In that carbon, beta oxidation is main to attach the carbon atom. Carbon atom. Coming to the next one. Here, no, Knoop's hypothesis. The scientist Knoop's how to hypothesis of the fat metabolism. That is called the evidence for two carbon structure. 36. So two carbon structure, unit metabolites of the fatty acids. These are fatty acids was suggested in famous experiment by the French noobs. French noobs, that is a fat metabolism breakdown the alpha. Beta oxidation is the cheap process of fatty acid. Beta oxidation is the very cheap process of the fatty acids. Degeneration in plants. The mechanism is well established with saturated fatty acid and unidentified unsaturated fatty acids. Beta oxidation takes place in mitochondria and involves segment removal of two carbon in the form of acyl coenzyme A. Okay, acyl coenzyme A. The beta oxidation is a very cheap process of fatty acid. Degeneration, degeneration of degeneration degeneration of the in plants. The mechanism is well. The mechanism is well. The mechanism is well established with saturated fatty acids and unidentified unsaturated fatty acids. These beta oxidation takes place in mitochondria and involves segmented removal of two carbon in the form of two acyl CoA molecule from the carboxyl and carboxyl and of fatty acid. This is called the beta oxidation. What is beta oxidation? Beta oxidation takes place in mitochondria. This reaction you can observe in the mitochondria and involves segmented removal of two carbon structure, two carbon in the form of acyl coenzyme. This one, acyl coenzyme A molecule. This form from the carboxylic and fatty acid. This is called the beta oxidation because beta carbon of fatty acid is oxidation during this process. The Knoops and hypothesis evidence for the two carbon unit metabolites of fatty acids was suggested in famous experiment French Knoop, in which dogs were fed derivatized fatty acid linked to benzene at the third and their end. Dog-fed derivated fatty acid with even number of carbon chain extracted phenylitic acid, that is the benzene plus 2 carbonic acid, whereas dog-fed derivatives fatty acid with odd number of carbon chains extracted benzoic acid. Benzoic acid is a one carbon structure. See, this is the carbon structure. See, you can observe this one. Even number carbon chain. Second one is the odd number carbon chain. The fatty acids, the fatty acyl-CoA, so produced again, re-enters the beta oxidation spiral at step 2. From the step 3, losing a further 2 carbon unit. This sequence continues until whole molecule is degraded. Each turn of beta oxidation generates one FADH2, one NADH2, and one acetyl-CoA molecule. One acetyl-CoA molecule. However, in the last turn of the spiral, two acetyl-CoA molecule will produced. Reoxidation of FADH2, NADH2 by the electron transport chain will yield 2 to 3 ATP molecules respectively. Thus, each turn of beta oxidation generate 5 ATP molecules. However, in the first turn, there is consumption of 1 ATP in the first step. Hence, in this turn, there will be net gain of 4 ATP molecules. This is a phenylalaninic acid. This one is a benzenic acid. Phenylalaninic acid, even number of carbons are there. Here, odd number of carbon is there. Fatty acid activated. Fatty acid activation taking place in the cytoplasm, where you can observe the activations are there in the cell where the cytoplasm is there. In that cytoplasm, activated place. Okay, fatty acids are activated to which enzyme? Acetyl coenzyme A. Which one? Acetyl coenzyme A is the that is the activated position. That is activated position. Acetyl CoA thiokinase or acyl co-thiosynthetase enzyme. The activated acyl CoA. What type of enzymes involvement are there? Thiokinases or acyl CoA synthetase. The reaction occurs two steps. The reaction occurs two steps. Two steps in the are required ATPs coenzyme A and Mg. Coenzyme A and Mg plus 2. Fatty acids react with the react with ATP to form react with the ATP to form acetaldehyde, which then combine with coenzyme A. Coenzyme A to produce the acyl coenzyme A. This one is the activated. This is the here three stages is there, no? First one, activation place is the cytosol, where you can observe the activation in the beta fatty acids. Beta fatty acids, how to phases to form. Here three phases to form is there. One is the activation of fatty acid in the cytosol. In the cytosol. Transport of fatty acid into mitochondria. This step to starting from to the in the cell, in that cell, in the cytoplasm. Cytoplasm to move from the fatty acid into mitochondria. Beta oxidation proper in the where you can observe this total process mitochondrial matrix. Where you can observe that one mitochondrial matrix. Fatty acids are oxidized by the most of tissue in the body. Brain, erythrocyte, and adrenal medulla cannot utilize fatty acid for energy requirement. Very, very important. Which one is not involvement is not there in that one? First one is the these total process you can observe in this one brain erythrocytes. You know, in the blood cells, erythrocytes are present. These erythrocytes and adrenal medullas cannot utilize these fatty acids fatty for energy requirement, because this is cannot requirement. And here fatty acid activation site is the cytoplasm. First activation site is the cytoplasm. Fatty acids are activated by acetyl coenzyme A, thiokinase, and acyl CoA synthetase. The reaction occurs in two steps and require ATP coenzyme A and magnesium atoms. The fatty acids react with the ATP to form acyl acyl adenylate, which then combine with coenzyme A to produce the acyl CoA. The two high energy phosphates are utilized since ATP is converted to pyrophosphate, that is called PPI. What is the name that one? Pyrophosphate. The enzyme inorganic pyrophosphate hydrolyzes PPI to phosphate. The immediate elimination of phospho in pyrophosphate makes this reaction totally irreversible. This reaction totally irreversible. The total reaction is irreversible. Thiokinase, this enzyme is the T different enzymes are there. One each are for short chain. Second one is the medium chain. Third one is the long chain fatty acid. The three different enzymes is involvement of the thiokinase. One is the short, second one is the medium, third one is the long chain fatty acid. Small chain fatty acid may also be activated by, which one is the activated? Small chain fatty acids may also be activated by the thiophorase enzyme. thiophorase enzyme and using succinyl coenzyme A also. Succinic coenzyme A also. Coming to this activation of fatty acid using this is a fatty acid. This is a fatty acid RCH2. Here the fatty acid or fatty acyl CoA, fatty acyl CoA produced again re-enter the beta oxidation spiral at space 2. This is a coming oxidation that again to beta oxidation. This sequence continues until whole molecule is degenerate. Whole molecule is degenerate. Each turn of beta oxidation generate one FDH and one NADH acetyl coenzyme molecule. This fatty acid to converted into the acetylenate utilizing enzyme thiokinase, some amount of ATP and magnesium ions to form to the fatty acyl CoA, or this one is called the acyl adenylate enzyme. This co-SHMP release to form the acyl CoA. The transport of acyl CoA into this process you can observe under to the totally found into the in cytoplasm only. Now these fatty acids now enter into this acyl now enter into the where in cytoplasm to mitochondria, in cytoplasm to mitochondria. Now the inner mitochondrial inner mitochondrial membrane is impermeable of fatty acids. These are the impermeable of the fatty acids. A specialized carnitine carrier system is there. Carnitine shelter operate to the transport activated fatty acids from the cytosol to mitochondria. This carnitine is the beta hydroxy alpha trimethyl aminobutyrate synthesized by the lysine and methionine in liver and kidney part. What is the stage? It occurs in 4 stage. Where you can observe this one, it occurs in the 4 stages are there. The acetyl transport into cytosol, that is a through CO donor to carbons mechanisms. This total is the occurs in the synthesis of the long. It occurs 4 stages. One is the acyl group of acyl CoA transferred to the keratin catalyzed by enzyme keratin acetyl transferase enzyme present on the outer surface of this enzyme. This is a CAT1. Okay, this small amount is a CAT1. This enzyme you can observe in the inner mitochondrial surface membrane. The acyl keratinine is transported across the membrane to mitochondrial matrix. This enzyme now across the inner mitochondrial to enter inner side of the membrane, that is the matrix side, by specific carrier proteins are there. This is the translocases. This protein is called the translocases. Next, another CAT2, carnitine acyl transferase 2 found on the inner surface of inner mitochondria. That is the outer surface of the inner mitochondria. This is one inner surface of the inner mitochondrial membrane. You can observe to the CAT2. Okay, this one is the CAT2 acetyl carnitine acyl transferase. This is converted into the acetyl carnitine to acyl coenzyme A. Now, these two transfer into the acyl CoA. The keratinine released return to cytosol for reuse by translocase. Reuse by the translocase. See, cytosol to keratinine transport system. This is cytosol. This keratinine, this is the mitochondria. This is the inner mitochondrial membrane. This is the mitochondrial matrix. This one is a mitochondrial matrix, total mitochondrial matrix. This how to acyl transport into cytosol to inner mitochondrial matrix through mal only. CoA is the donor. Here CaO is the CoA is the donor of two carbon fragment to the increasing length of fatty acid at every step. It itself is synthesized from acetyl CoA through fatty acids or synthesized in the membrane of endoplasmic reticulum here of the cytoplasm. Endoplasmic reticulum of the cytoplasm. The acyl CoA itself is formed within the mitochondria. This as a result of oxidation of pyruvic acid, fatty acid, and amino acid. Since the acyl CoA is non-diffusible across the mitochondria membrane, it transfer to the cytoplasm involves a acyl group shelter. That one is the acyl group shelter. The inner mitochondrial, this one, acetyl CoA. Now, the acetyl CoA first react with oxaloacetate to form citrate in the presence of the enzyme citrate synthetase enzyme. First reaction, this is the production of the Krebs cycle. The citrate can diffuse out of the mitochondria with the help of tricarboxylate carrier. This is the citrate react with the CoA and ATP cytosol to produce the acetyl and oxaloacetate in the presence of an enzyme called citrase lysase. Citrate CoA ATP, acetyl CoA, oxaloacetic acid plus ADP plus PI. Since oxaloacetate cannot diffuse into the mitochondrial, it enters the mitochondria in the reduced form the malate. The malate is oxidized to regenerate the oxaloacetate. This is a now to form this one. The acyl CoA undergo carboxylation to produce the malonly CoA. Wiki scientists discovered bicarbonate needed. The total mechanism citrate, isocitrate to involvement is present. Beta oxidation proper. One is the oxidation. Each cycle of beta oxidation liberating two carbon unit acyl CoA occur in the synthesis sequence of four reactions. One is the oxidation position. The second one is the hydration. A third one is the oxidation. Fourth one is the cleavage. Fourth one is called cleavage. What is the first oxidation? Acyl CoA undergoes Dehydrogenation by the flavin adenine dinucleotide, that is the flavin enzyme, FAD, depend flavoenzyme, acyl CoA dehydrogenase enzyme. A doubled bond is formed between alpha and beta carbon. That one is called 2 and 3. That is called the 2 and 3 carbons. This is called the oxidation. The second one is called hydration. The second one is called the hydration. Hydration, enoyl CoA hydrates, brings about the hydration of the doubled bond to form beta hydroxy acyl CoA. That is the beta hydroxy acyl CoA. The third step of the oxidation, beta hydroxyl CoA dehydrogenase catalyze the second oxygen and generate of the NADH. The product formed beta keto acyl CoA. This is the third one. The last one is the cleavage. The final reaction in beta oxidation is the liberation of a 2 carbon fragment. Acyl CoA form. Acyl CoA. This occurs by a thiolytic cleavage catalyzed by the enzyme beta keto acyl CoA thiolase or thiolases enzyme. See, this is the carcinate auto to transfer fatty acid utilizing some amount of ATPs and thiokinase the enzyme to form the acyl CoA. These are to form to the cytosol of this cell. This cytosol of the cell acyl CoA now enter into the now enter into the mitochondria. See, this is the mitochondria. Now inner mitochondria, these acyl to form and for to move that one. Here acyl coenzyme, acyl co dehydrogenase utilizing the FAD to loss of hydrogen to converted FADH2 and to release the 2 ATPs to form to the trans-enol CoA. Trans-enol CoA enzyme to form. This trans-enol CoA to adding some amount of water to form to the beta oxidase enzyme. Now beta hydroxylase CoA. This one be the utilizing the from NAD plus 2 and to form beta keto acyl CoA. Beta keto acyl CoA. This is called the to split in two. One is the 2 acyl CoA. One is the CoA acyl CoA. Thiolase acyl CoA to split in this one. The total oxidation Plametally CoA plus 7 CO CoASH plus 7 FDP, 7 NADP plus 7 H2O to form acetyl CoA plus 7 FADH2 and 7 NADH2, 7H plus Plamotylyl CoA undergoes 7 cycles of beta oxidation to yield 8 acetyl CoA. Acetyl CoA can enter citric acid cycle and get completely oxidation for the CO2 and H2O. This is the energetic beta oxidation. The total, what is the energetic table of the beta oxidation? Beta oxidation, 7 cycles. Here, 7 FADH2 oxidized by the electron transport system. that is oxidation is no. That one is called the electron transport system. Electron transport system is each FAD2 gives to the two ATP molecules. To your 2 FADH to total ATP, 7 into 2 is equal to 14 ATP yield to form. Here, the 7 NADH is equal to 3. 7 into 3 is equal to 21. NADP into is equal to 3 ATPs. FADH2 is equal to 2 ATPs. In the above one, 7 into 2 is equal to 14. That's where the ATP yield to produced 14 ATP molecules. In this one, 7 NADH2 to produce to the electron transport system oxidize each NADP liberated from the 3 ATP. 7 into 3 is equal to 21 ATP. This is the first step of oxidation. The second form of 8-acyl-CoA oxidized by citric acid cycle. That citric acid cycle meaning Krebs cycle. Each acyl-CoA provide 12 ATPs. The total 96 KTPs. And total energy from one molecular Planotial CoA 131, energy utilized for activation -2, formation of how minus planotic formation, no, that's where 131 -2 is equal to net yield of oxidation of one molecule plamatylate is equal to 129. Okay, the regulation of the beta oxidation. How to regulate this process? What is the regulations are there in this process? The availability. Main is the regulation is called the availability of free fatty acid. FFA is called the free fatty acid. Regulate the net utilization through beta oxidation. Here, the level of the free fatty acids controlled by how to regulations are there in the fatty acids. How to free fatty acid regulates controlled by the glucagon insulin ratio. Glucagon and insulin ratio. Glucagon increases FFA level, free fatty acid level, and insulin as the opposite effect. Insulin as the opposite effect. Here, CAT is a regulatory of entry of the fatty acid into mitochondria. Where is entry into the mitochondria. Melanoyl-CoA inhibit CAT1 activity. CAT1 activity. Thus, during the deneosynthesis of fatty acid, the beta oxidation is inhibited. The beta oxidation is inhibited. Sudden infant death syndrome, SIDS is a unexpected death of the healthy infants, usually overnight, due to deficiency of medium chain acyl-CoA dehydrogenase enzyme. Glucose is the principal source of energy soon after eating or feeding babies. Soon after eating and feeding babies. After few hours, the glucose level and its utilizing decreases, and the rate of fatty acids oxidation must simultaneously increase to meet the energy needs. Energy needs. The sudden death in infants is due to blockade in beta oxidation caused by the definition in medium chain acyl-CoA dehydrogenase in this one. Next one, Jamaican vomiting sickness. A characteristic by severe hypoglycemia, vomiting, convulsion, coma, and death. It is caused by eating unripe fruit contain an unusual toxic amino acid, hypoglycemia A. This inhibits the enzyme acyl-CoA dehydrogenase and beta oxidation of fatty acid is blocked, leading to the various complications. Various complications. Abnormality in transport of fatty acid into mitochondria, defects in oxidation leads to the deficient energy produced by the oxidation of long chain fatty acid. What are the features of in this one? Hypoglycemia, hyperammonemia, weakness, liver disease, acyl carnitine accumulates when the transferase or translocase is deficient. Dietary supplementation of carnitine improve the conditions. What is the oxidation of odd chain fatty acid? Odd chain fatty acids just like similar to the even chain fatty acid. At the 3 carbon unit, propionyl-CoA is produced. Propionyl-CoA is converted into succinyl-CoA. Succinyl-CoA is an intermediate in TCA cycle, the Krebs cycle. Intermediate in the TCA cycle. This is the principal propionyl-CoA is gluconeogenic. That is the gluconeogenic. Coming to the conversion of the propionyl-CoA to succinyl-CoA. Propionyl-CoA is the carboxylated to dimethyl melanyl-CoA by a biotin-dependent carboxylase. Biotin ATP is utilized in this step. Methyl melanoyl-CoA racemase act upon dimethyl malonyl-CoA to give L-methyl-malonyl-CoA. This reaction is essential for the entry of this compound into metabolic reactions of the body. The methylmalonyl-CoA mutase catalyze the conversion of L-methyl melanoCoA. This is the total conversion of succinyl-CoA to propionyl-CoA. See, this is the propionyl-CoA to succinyl-CoA. Okay. Now, this is the total. This is the total fatty acids lipid metabolism is completed. The total portions is in the beta in your syllabus, beta and alpha oxidations only are there. These are the total completed. Now we will discuss about to the minerals and what are the minerals, what is type, how many types of minerals are there, and mineral salts, macro minerals, micro elements, minor nutritions, that is the macro molecules, mineral micro elements. How many minerals are involvement is there? These are things we will I'll start that one for that class. Okay. One second, I'll share my PPT. Now, mineral after next class we will discuss about the minerals. Now we will discuss about to the enzymes and coenzymes. One second, I'll share my PPT. Now I will share my PPT. Now the topic of the fatty acids are totally completed. That is the lipids, lipid metabolism. These all are completed in the lipids, simple lipids, compound lipids, simple lipids, uh, compound lipids, derived lipids, biological significance, fat metabolism. These all things are completed. Now we will discuss about to the enzymes and coenzymes. Very important topic, enzymes and coenzymes also. First, what is the enzyme? What is the enzyme is? Enzyme or the biological, you can identify the definition of the enzyme, history, importance, properties of enzymes, classification, different class of enzymes, nomenclature, individual classes, individual classes. These all are the importance of the, you can study the enzyme. The study of enzymes is called enzymology. What is the study of enzyme is called enzymology or enzymology. You can study the enzyme, each and every physiological, the enzyme is the most important topic. These enzymes involve in all the functions of the total reactions, reactions under control to the enzymes. That's why the study of enzyme is called enzymology. Who studied the enzymes? That is called the enzymology. The main is the enzyme. Another one second. One second, I'll explain. 361. Mm. Uh, this is the enzymology. Into the group, the main is the enzyme is the food synthesis by plants is generally stored in the form of large complex. Coming to the next point, definition. I will explain to the first definition of the enzyme. In the enzyme, what type of all are there? This is the what is the enzyme, this one. First we will discuss how to enzyme, who scientist is the enzyme. Food, main important of the definition is the enzyme. The food synthesized by plants is generally stored in the form of large complex molecule which are usually insoluble. The conversion of these complex substance into simple, soluble, and diffusible form is known as digestion. That name is called the digestion. This makes the organic compound easily available to the different parts of the plant. The several chemical reaction with the living cell during digestion can proceed in an orderly fashion only in the presence of certain specific substance or organic catalytics called enzymes. What is the enzyme? The several chemical reactions within the living cells during digestion can produced in an orderly fashion only in the presence of certain specific substance or organic catalysis is called the enzyme. At their simplest, enzyme may be considered as catalysis produced by living protoplasm. The history of enzyme begin with the work of Duberfant in 1830, who prepared malt extract from germinating barley seed. The starch was found to be hydrolyzed into sugar by the malt extract. Another scientist, Payan and Persoj, in 1833, extracted the enzyme diastase from the malt extract. Another one, Pocher scientist, in 1857, demonstrated that the living yeast cell were responsible for alcoholic fermentation. Thus, two types of ferments or catalysis were distinguished: unorganized ferments. Unorganized ferments like diastase, organized ferments like living yeast, etc. The term enzyme was later proposed Kuhne. The scientist name is Kuhne in 1878 for non-living unorganized ferments. The modern era in study of the enzyme began when Buchner scientist in 1897 accidentally discovered that a juice extracted from yeast cell brought about fermentation of sugar in the same way in which is the living yeast cell is died. When the juice was boiled, it lost its power of fermentation. The extract was found to be complex enzyme is known as zymase enzyme. Enzyme are basically protein molecules, but this is all are the protein molecules. Many of them have non-protein thermostable group attached to the protein molecules. Enzymes differ from conventional catalysis in that they are proteinous and so are profoundly altered by any changes in their environment which affect their chemical or physical properties. They also differ in their remarkable specific action, which is probably a control of their protein structure. Borderly enzyme carry out two important functions in living organisms. In the first place, they are able to two different parts. One is the biological catalysis. The second one, biological enzymes or biological catalysis. Biological catalysis accelerates the rate of chemical reaction. This is a chemical reaction capable of performing multiple reaction or multiple recycled. Finally, distribution of reactant and product governed by the equilibrium process properties. This is a biological catalysis. The enzyme are biological catalytic proteins. All enzymes are proteins. A few RNA expectation. All, even when you can ask the enzymes are, all enzymes are proteins, a few RNA expertise, orders of magnitude faster than chemical catalyze act under mild condition, temperature and pressure, highly specific, tightly regulated. Okay, now I'll explain now what is the history by Berzelius in 1836 coined the term catalysis. This is a Greek word. Catalysis to dissolve. In 1878, Pune, first scientist name identify the this part is called the Pune used the word enzyme. This also Greek word. Enzyme also Greek word. This is called yeast to indicate the catalysis taking place in the biological system. Next one, isolation of enzyme system from the cell-free extract of yeast was achieved in 1883 by Buchner scientist. He named the active principal name is called zymase. Later, this contained mixture of enzyme. The butcher scientist that is called, the name is called zymase. Later on, this one is called mixture. Contain a mixture of enzyme which could converted sugars to alcohols. These all are converted into the sugars to alcohol. In 1926, James Sumner scientist, James Sumner first achieved the isolation and catalyzation of enzyme urease enzyme in jack bean. The James Sumner first cultivated, achieved, so not cultivated, achieved the isolation, crystallized form of enzyme. First crystallized form of enzyme to achieved the scientist, Sumner scientist, that is called urease from the jack bean. This is a history. What is the importance of the enzyme? Enzyme critical for every aspect of cellular life. Enzyme, the cell shape and motility of cell, surface receptors, cell cycle, metabolism, total transcription, protein synthesis, hormone releases, muscle contractions, protein synthesis. These all are the prospectives involvement or the importance for the enzyme. Okay. What are the properties? How many properties are there? This is a vital role for chemical reactions to occur in the cell. All chemical reactions under in the cell control the enzyme. The breaking, forming, and rearranging of the bonds on substrate, that is the reactant. Modified substrate now is a product often performs different task. Consequence, transformation of energy and matter in the cell, cell to cell, intracellular communication, allow for cellular homeostatic to persist. This is the properties of enzyme. Classification of enzyme. One second. Classification of enzyme. The older classification of enzyme is based on the basis of reactions which they catalyze. May earlier authors have classified enzymes in two categories. One, how many classification enzyme can be classified using numbering system defined as the enzyme commission. This system consists of four-digit number. That is called the four-digit number, which classified based on type of reaction the enzyme catalyze. One example, the hydrolytic enzyme. Authors classified in two groups. No, the classification is the two groups. One is the hydrolyzing enzyme. Second one, desmolizing enzyme. Hydrolyzing, desmolizing. Other classification of enzyme, three groups. Hydrolyzing, transferring enzyme. Third one, desmolizing enzyme. In the first classification, transferring enzyme are included in the hydrolyzing enzyme, since some of them are known uh, to act as transforming as well as hydrolyzing enzyme. What is the different class? Oxidoreductase, transferase, hydrolyzase, lyases, isomerization, ligases. These six classes of the enzyme. See, first one, oxidorode transfer electrons. Transferase transfer functional groups between molecule. Hydrolase enzyme break bond of adding O2, H2O. Lyases reaction from doubled bond. Elimination. Lyase is the splitting of the enzyme. Isomerase intramolecular rearrangement. See, you can by heart. This is the most important. These are all the basic points. In case you can study the molecular biology or entomology or physiology, these things also most important. Oxidoreductase. Oxidoreductase transfer electrons, redox reactions. Where the oxidoreductase is there? To transfer one molecule to another molecule electrons. The second one, transferase enzyme. Transferase enzymes here, uh, transferase enzyme transfer functional group between molecule. Transfer functional between molecule. Next one, hydrolase. These break the bond by adding water. Lyases. This is the elimination. First, I'll explain today what is the hydrolyzing enzyme. Nomenclature we will discuss about later. Transferase hydrolases enzyme. Okay. See, now these are things we will discuss later. Okay. This is a different class, no? Oxidoree, trans, hydrase, ligase, isomerase, ligase. Now I will explain to this one, hydrolase. Coming to the hydrolase enzyme. See, this one is a hydrolase. Okay. The hydrolase enzyme or hydrolases, both are same, don't confuse. The hydrolyzing enzymes or hydrolases catalyze reaction in which complex organic compounds are broken into simple compound or broken organic compounds, or complex organic compounds are broken into simpler compounds with the adding of the water. 1 second, I'll take the pen. Yes. See, in the simple or complex organic compounds are broken into simple compound. How to broken? Some amount of adding water. Amount of adding water. These are subdivided into 4 groups. How many groups are there? This one? Totally 4 groups. These are the 4 groups. Here, groups on the basic, the nature on the substance. First one is the first one, carbohydrates. The first one, CH. CH meaning carbohydrates. 1 second. Where is this? Mhm. The first one is called carbohydrate. Most of the polysaccharide, disaccharide, or small oligosaccharides are hydrolyzed to simple compounds. What is the carbohydrate? Polysaccharides, disaccharides, or small oligosaccharides are the hydrolyzed to simpler compound. Example, hexose, pentose under influence of these enzyme. Hexose and pentose. Hexose sugars and pentose sugars is the example. Here, under influence of this enzyme, 1 second, I'll write here. See, sugars, sucrose, sucrose, sucrose to converted into glucose plus fructose. Glucose plus fructose. What is the enzyme? Sucrase. CRASE. The final, you can write down the A's. In the nomenclature time, I'll explain each and every enzyme. The final, you can write down the age. ACE. Nomenclature. Most of the enzyme name different suffix. A's to the name of the substance. Sucrase, maltase, lactase, okay, maltase, lactase, these are that one. That is the structure of enzyme, the primary structure, secondary structure, tertiary structures are there. The protein is based of the number sequence amino acid in the protein chain. This determined covalent bond, amino acid in the chain. This is a primary. The secondary is the concern the manner with the peptide chain is folded and the nature of the bonds with the stabilized structure. This is the hydrogen bonds are formed between the carboxylic amide is the polypeptide chain secondary. Tertiary folding, bending of the protein chain. This occurs due to the interaction between the side chain of the amino acids is a tertiary structure. That is the structure protein. Nomenclature. Most of the enzymes are named by adding the suffix ase to the name of the substance. Sucrase, maltase, lactase. The name of some enzyme indicate the nature of the reaction, which is they take part oxidase, dehydrogenase, etc. Generally, the name of the specific substance is also mentioned along with the such names. Okay. There are some enzymes which are named in an arbitrary was such as the pepsin, trypsin. This is the arbitraries. Okay. This is a isoenzyme, alloenzyme. We will discuss later. Isoenzyme, alloenzyme, multi-complex of enzyme. This is the first classification. Sucrose, sucrase enzyme to convert glucose, fructose. Next one, maltose. Maltose converted into glucose. This is a maltase enzyme. Okay. Raffinose. Raffinose converted into the fructose plus melibiose. Okay. Next one, cellobiose. Cellobiose, the utilizing enzyme sucrase converted into glucose. Next one, lactose. Lactase enzyme converted into glucose plus galactose. Lactase, glucose plus galactase. Next one, melibiose. The enzyme melibiase enzyme converted into same, this one also, glucose plus galactose. Next one, starch. Starch amylase enzyme. Starch, what is the found in starch? Amylase. Amylase enzyme. Amylase enzyme. Starch converted into maltose. Starch is a maltose. Cellulase enzyme, cellobiose. Inulin enzyme, inulin enzyme to converted inulin in utilizing enzyme inulase converted into fructose. These are all the carbohydrates. Okay. This is the first one. Carbohydrates in the hydrolase. First one. The second one is esterases. The second one is called the esterases. Esterases, these enzymes catalyzed the hydrolysis of substance containing ester linkase. Example, fat. Esters is called fat, pectins. Fat ఒకటి, fat and second one is the pectins etc. into an alcoholic acid, acidic compounds also ester. Here fat, this fat lipase enzyme utilizing converted into glycerols. These glycerols plus fatty acids. Glycerols plus fatty acids. Next one, phosphoric acid. I'll write here. Phosphoric acid ester converted phosphatase enzyme converted into phosphoric acid. Phosphoric acid plus other compounds. Next, lecithin. Lecithin utilize lecithinase fatty acids. That is the fatty acids plus lysocithin. Next one, lecithin divided to fatty acid plus choline plus glycerophosphate. These are all the esterases enzymes. Here many are there. Glycerophosphate, glycerol phosphoric acid. Next one is the adenosine triphosphate, adenyl pyrophosphate converted into adenyl acid, AMP, phosphoric acid, fructose 1,6-diphosphate. Here myosin exodiphosphate, fructose 1-phosphate, phosphoric acid, phytin, inositol plus phytosyl. These chlorophyllase, phytol plus chlorophidyl, pectin, pectidase enzyme, pectic acid plus methyl alcohol, phytin, phytase enzyme, inositol phosphoric acid, nucleic acid, ribonuclease, deoxyribonuclease, component nucleic acid. These all are called the esterases. The third one is called the proteolytic enzyme. What is the third one is called proteolytic. The hydrolysis of protein into that is called peptones, polypeptide, and amino acid is catalyzed by these enzymes. Protein to peptones. Polypeptide peptidase enzyme converted into amino acid. Last one is the amidase. They hydrolysis amide into ammonia and acid. Urea converted urease enzyme. The pepsine last one is the urea. Urea is the urease enzyme. It converted into the ammonia and carbon dioxide. Next one is the asparagine enzyme, ammonia and aspartic acid. Okay, aspartic acid. Most of the desmolizing enzyme are the enzymes of respiration. Enzyme concerned in the synthesis of amino acid are also included in this group. A detailed account of such enzymes will be found in the chapter will be found in the chapter on respiration and next one is the nitrogen metabolism. What are that one? Kinases, desmolarization, kinases. Second one is desmolase, dehydrogenase, and oxygenase, transminase, carboxylase, hydrase. Next one, flavoprotein, peroxidase. So these are the uh, these are the properties. IUPAC system of classification. This is the most importance of the enzymes. Now we will discuss about to the enzymes. Before that one second. See how to oxidor nomenclature, this one. IUCNB rules, the IUCNB rules, enzyme nomenclature, IUBNC rules, the rules of IUBNC. 45. In the recent past, the International Union of Biochemistry has given a more rational classification of enzymes based on the reaction types and reaction mechanism. The major features of the IUB systematic of the classification of enzymes are follows that one reaction and the enzyme catalyzed them. They are divided into 6 classes. How many classes are there? They divided into how many classes? 6 classes divided into 6 major classes, each with how many? Each and with one 4 to 3 subclasses. Each 1 to 4 to 3. The enzyme name has 2 parts. The first name of substrate. The second name is called substrate or substrates. Substrate or substrated. The first name is substrate. The first is the name of substrate or substrates. The second ending is in indicate reaction catalyzed ase. You can write down ase, substrate and ase. That is the reaction catalyte. Each enzyme has systematic code number. This is the EC is called the systematic code number. The first digit denotes, see, 3221 is there, no? The first number digit denote the class. This one is the first type of general reaction, that is called the class. The second number is called subclass. The third number subclasses. And fourth one is for the particular enzyme named. Thus, for the enzyme code, that is called the enzyme systematic code number. The EC value, example 3211. 3 is the denote to class 2 transferase. 3 is the hydrolase enzyme, type of general. The second one is the subclass enzyme glycosidase. Third one, subclass enzyme reaction hydrolase or glycerol group. The last one is indicate specific enzyme alpha amylase enzyme. In case I'll give the another example also, 2.7.11. Okay, this 2 is denoted transferase. This is a transferase. This denote the class. This is the class is a transferase. The 7 subclass transfer of phosphatase enzyme. The third one second one is the subclass. Third one is the subclass and alcohol function the phosphate acceptor. The fourth digit one indicate exokinase. The one is the exokinase 1. The additional information, if the nature of the reaction is to be further clarified, additional information may follow in the parenthesis. Malai enzyme which oxidized L-malate to pyruvate in the reaction. L-malate plus NAD plus is equal to pyruvate CO2 plus NADH plus H plus is thus also known as, that is called 1:1.3.7. That is called L-malate. That one is called the L-malate NAD plus oxidary reductase carbolase enzyme. This is the total nomenclature of the enzyme. See, we will stop this much only. Next week, we will continue of the total nomenclature of the enzymes. Okay, the nomenclature and the different types. You can observe the total enzymology and like this only. Now we will stop the enzymology in the last yesterday class and today class. Yesterday we will discuss about to the things. What is the lipid metabolism is completely totally completed. What is the what one? Lipid metabolism is totally complete. The next today we will start about to the enzyme and enzymology. Okay, bye. We will meet on next week. That is the complete of the systems enzymes. We will complete in that day. Okay. नहीं आ रहे हो आ रहे हैं। आ रहे हैं।